Hello students, welcome to Lab 11. We're talking about fermentation. This lab is mainly going to be going over the basis of fermentation. Okay, so I don't think this is going to be too long of a lab. Okay, so first of all, fermentation happens in the absence of oxygen, right? Fermentation is different from anaerobic cellular respiration, right? Fermentation is an aerobic reaction, but it doesn't require oxygen, but it's different than anaerobic respiration in that it only goes through glycolysis. It doesn't go through the Calvin cycle. So there's many different types of fermentation. In this lab, we'll focus on ethanol fermentation, right? So we use the bacteria or the mold yeast Saccharomyces cerevisiae. This is used to make beer, bread, wine. So they go through the process of ethanolic fermentation to make CO2, ethanol, and energy, basically making 2 molecules of ATP. It's clear that fermentation is much less efficient than cellular respiration or anaerobic respiration, as it produces much less ATP. However, it's the only method available for these organisms that can't get oxygen in its environment. All right, so we have the fermentation experiment. We're going to be using 4 different test tubes, right? We have glucose, lactose, sucrose, and distilled water, right? So which sugar do you hypothesize will be used most efficiently to do fermentation? So glucose is a monosaccharide, lactose is a disaccharide, sucrose is a disaccharide. So sucrose has 12 carbons, lactose also has 12 carbons, sucrose and lactose are the same chemical formula, but lactose may be a little bit more difficult because lactose is more chemically bound because it has galactose and glucose subunits, right? Sucrose is formed with glucose and fructose. It's refined from table sugar. So it's up to you. The better the fermentation, the bigger the bubble will be in the tube because the gas that will collect is CO2. Right? If you look into the formula, what gas is being produced? It's CO2. It's the bubbles that is being produced. So if you look in the table that I have for you in the attendance or in the lab assignment, it'll show you what the initial results and the final results are for the experiment. All right, and then you should be able to answer the questions that I have for you. Make sure to answer all of those questions. And then I have some review questions for your lab practical. All right. I think that is it for this time. Yes. All right, if you have any questions with this one, make sure to ask me. All right. Keep in mind to keep working on your lab reports. Right? Keep working on those. They're going to be due fairly soon. I believe I said November 2nd, right? So now would be a good time to work on those, considering that this lab is rather short. The next two labs are going to be somewhat long. Okay. All right, I'll see you later.